I'm going to try and make up some time here. I know we're coming up on our break, but I wanted to start just by thanking Rajiv and the organizers. This is my first time to ICRISAT, first time to India, and it's one of the best conferences I've been to in a while in terms of the quality of the interactions with the people. In fact, when I saw who was coming, ordinarily I would speak about genomic selection at a meeting like this, but with so many good people, I decided, well, why not try something totally different? So actually, I won't be speaking about genomic selection at all today. Before I get into the main topic, I wanted just to tell you a bit about what I'm doing professionally, because I've transitioned recently from working at Cornell and working closely with the Gates Foundation. I'm trying to become a potato breeder now, working at the University of Wisconsin, where potato production is an important part of the agricultural economy. Actually, the breeding program at the university started in the 1930s, so it's one of the oldest breeding programs in the U.S., and we test about 50,000 genotypes each year. And of course, I'm hoping to do a lot of the usual tricks that we're all interested in here in terms of trying to take potato breeding into the modern genomics era. It's a very traditional phenotypic selection-based program at the moment, but has some interesting preliminary results about GWAS and using GBS markers and genomic selection and how do we make all this work in autotetraploids. But having just started this a few months ago, I'm not in a position to talk about it today, so perhaps I can come back in a couple of years and share some of that story. What I wanted to talk about was a new release of a software package, which I think will be of interest and perhaps of use to some of you in the audience. We've been talking about, several people have talked about consensus maps, and it's come up in several of the presentations, and so this is a new R package that produces consensus maps. Just so we're all on the same page, what is a consensus map? It's a genetic map that's constructed across multiple biparental populations. The reason why you would want to do that is because in any given biparental population you have a limited number of markers that are segregating, and so by integrating across multiple maps you can get a higher density map. And this is useful. It's an enabling technology for a variety of reasons. One, to help anchor, to have something to anchor scaffolds to if you're making a whole genome sequence, when you're analyzing your germplasm for LG patterns or to do association mapping, it's useful for that as well. I got involved in this through an association mapping project in Barley. We heard a bit actually from Frank yesterday, I think it was, about barley food uses. It's not just about animal feed and malt for beer. So there was a project in the U.S. as well trying to look at how do we breed barley for food applications. This is part of my PhD work when I got into this at Washington State University. And some of the traits that were being evaluated there are, for example, the kernel hardness, which is the force to crush a kernel of barley, and it's related. It has a relevance for the milling properties, like the particle size distribution and water absorption and whatnot. Polyphenol oxidase, example of another trait that's related to the enzymatic darkening. So if you make barley noodles, you don't want them to turn brown, after you cook them. Anyways, I include these two results just to show a contrast, typical contrast of how some traits can have really different genetic architectures in a population. So this is a group of 370 elite barley lines from the U.S. Pacific Northwest breeding programs. The PPO association mapping, we have a huge signal, and lo and behold, it's at a location where we knew there was already PPO genes in barley. So that's not too surprising. What was surprising was that for the hardness, we were not picking up really any strong signatures, even though we knew that there was a so-called hardness locus. That's what the HA stands for. There's a so-called hardness locus in barley, well characterized in wheat, but also to some extent in barley. When I tried to look in more detail in this region, I learned that we actually had markers tagged to the 3 specific loci in the in genes, or very basically, in very good LD with genes. And there are 3 genes. They go by the names of GSP, HIN A, and HIN B. The HIN stands for hordoindoline. It's a type of protein in the endosperm. Anyways, the important thing I want you to take away from this figure is to compare, say, the results of the association mapping and looking at the relative order of the markers for this locus compared to what we know they are in the physical map. So okay. So from the clone sequence, we know that the most distal marker is HIN B, followed by HIN A, and then the GSP. And if you look in the consensus map that was used in the association mapping, actually the order of the markers is not consistent with the physical order. The most distal marker was the GSP in this set, then the HIN B, then the HIN A. So it led me to kind of wonder a bit more about where was this consensus map coming from that was being used by the Barley cap team, and it dates back to this paper in 2009 where they published a consensus map that was developed by merging linkage maps from 4 Barley populations using a software called MergeMap. And I know a number of you have used MergeMap. I've seen papers from people in this audience, so you have some familiarity with it, but just to get us all on the same page, the way MergeMap, it's a graph-based algorithm, and it uses a graph representation of linkage maps. So just to kind of show you that here's the way of representing linkage maps for 3 of the populations. And each of the circles represents a bit of markers that are co-segregating. So all of these markers here are co-segregating in population 1. And then there's an arrow from one bin to the next bin, and the number on the arrow tells you the distance in centimorgans. What you should take away from this is to see that in population 1, all of the markers that were in this hardness locus, which I've got the HIN A and HIN B GSP names there, they're all co-segregating in population 1. In population 2, there was a single recombinant actually, which allowed us to determine that in the linkage map for that population, the HIN B marker was distal, which is consistent with the physical map that I showed you a moment ago. In population 3, again, all of the markers are co-segregating. So we have relatively little information from the linkage maps about what the relative order of the markers are at this locus. The one piece of information we do have is entirely consistent with the known physical map. But curiously enough, when you put this together in MergeMap and you look at what comes out of it, you get a distortion of the order of these markers. And that's what I had noticed in that association mapping. And so here's what the consensus map looks like from the MergeMap software. Again, the most distal set of markers is this group right here. And those are coming from this information over here, but they're showing up distal for reasons that don't really make sense initially. There's actually an intermediate stage in this whole MergeMap process, which is a branch type of graph structure called the consensus graph instead of a map. And you see how there's branches there. Now, that is maybe something that many of you aren't familiar with. What does a consensus graph mean? And so here's a slide just to try and explain that. It's an idea that goes back about 10 years, actually. A group at Cornell kind of pointed out that if you allow for branches in these types of graph structures, you can represent both what you do know and what you don't know about the relative order of markers. So the observation here is that when you have multiple populations, some of the markers that are private in the 2 populations, their relative order is not necessarily known precisely. So for in this example here, population 1, we have A, then C, then D, and that's captured by the left branch in the graph structure. And then population 2, we have A, then B, then D, and that's captured by the right branch. So we don't know from this particular diagram which is the relative ordering of C and B. It's indeterminate. And you could, of course, use the actual distances in the 2 populations to try and make a guess there. But if you only strictly use the implications from the 2 populations separately, this graph exactly represents the knowledge that's present. All right, so just to come back again to what the output from MergeMap looks like, you can see here's this graph structure here. In principle, the graph should have an exact equivalency with the information from the linkage maps, but it doesn't. Again, even in the graph structure, you see this set of markers showing up as being more distal. And again, that information is nowhere present in the linkage maps. So there was some additional simplification that was made when that consensus graph was generated, and that's what's leading to the kind of spurious ordering in the consensus map. All right, so when I realized this, I decided it was time to write some software that would not have this problem. So I created this package called Dagger, which was designed to create the correct, exact consensus graph, and published an article about it that describes its application to this very same Barley set of populations. So on the left, you see the exact consensus graph for what this distal region should look like, and you can just see how much more complicated and branched it really is compared to the output from the MergeMap software. There is a type of simplification that can be done where you can prune away extra arrows that aren't necessary from the perspective of making the implications about the relative order, and that's called the transitive reduction. So it creates a somewhat simplified picture, but it's still quite bushy compared to what you might expect from the merge map. So here again, here's a comparison side by side. The Dagger output, this is the correct consensus graph for this region. This is a simplified version, which has some spurious implications. In particular, if you this group of markers here, which shouldn't be here, what we know from the physical map, here is where it shows up in this Dagger graph. And you can see it's kind of floating. It doesn't actually, its order is not determined relative to these other set of markers, which is correct. We don't know the relative order based on the individual linkage maps. So it kind of illustrates that it's working properly, but the graphs are useful for capturing the information, but they're ultimately not that practical in terms of, say, doing any kind of analysis with respect to genetic distance. So we do want to collapse them down to a consensus map, a linear structure, and that's where you have to now introduce some approximation and think about an error minimization problem. And the insight I had was that actually by framing the error in a certain way, it allowed for a type of computer science optimization known as linear programming. The advantage of doing it is that it's very scalable, so you can do tens of thousands of markers, dozens of populations. It's a type of algorithm which scales to very big problems. So just conceptually, the type of error is a sum over the populations, and then within each population, the sum over the pairs of markers, and then what is the distance between the pair of markers in the linkage map and the distance between the pairs of markers in the consensus map, and take that absolute value, sum that all up, and that's your error. And that can be converted into a linear programming problem. One other thing to point out, though, is that just by minimizing the error, you aren't necessarily going to preserve the order of the markers that are present in the linkage maps. In particular, if all of your linkage maps are all consistent with respect to the marker order, you'd like that order to be preserved in the consensus map, and that is not a property that linear programming will offer if you only include error minimization. You have to add additional constraints to say that I want to force, let's say in this example here, I want to ensure that marker B, which is, say, comes after marker A in this linkage map, I want to make sure that property is preserved in my consensus map. So you do that by putting this inequality that says the position of marker B is greater than or equal to the position of marker A. So you generate all these inequalities for each one of these linkage maps. And if the inequalities are consistent across the linkage maps, then you have a linear program that you can solve. And here's an example of, in the case of these 4 Barley populations, actually miraculously the 4 linkage maps have no inconsistencies in order. They're entirely consistent. So the Dagger software runs very well, and here's a comparison showing the error on a kind of average per marker pair, what the average error was with the Dagger versus the merge map. And you can see that we're getting an improvement in 1 to 2 centimorgans on average across all the chromosomes. So we're getting a better consensus with our linkage maps compared to what the merge map software offered. So that was fine and dandy. I guess one other thing, just to show you, practically speaking, what does this mean? You can get some better results when you do the association mapping. I took the original figure I showed you and I just flipped it upside down so that you can kind of compare. It's the same association mapping signal. I just plot the points at different points on the horizontal axis because I've changed where I plotting them on the consensus map. And you get a nice collapse of all of those significant markers down to that one very small locus, which is a more realistic representation based on what we know from the physical map. Okay, so it usually is the case that you're always going to have, usually you're going to have conflicts in the order of markers across the different populations. It's rare that you would generate linkage maps for a bunch of populations and then it would be entirely consistent because you're always dealing with small populations, you have genotyping errors, and just the limitations of these ordering algorithms, the linkage maps are not going to necessarily be consistent even when you have one true physical structure. I mean, there are, of course, the biological differences in order, but let's say even if you had one biological order, you would still have differences in linkage maps. And the Dagger software couldn't handle that situation, which made it somewhat limiting in terms of its application. And I received a lot of emails after I published this and people saying, well, we're trying to use it, but it doesn't work because we have inconsistencies in our linkage maps, which I knew that when I published the paper. So it spurred me to try and find a solution to that problem. And that's what this LPmerge software does, is it overcomes now that original limitation because it resolves the marker order conflicts. So here's an I'm not going to describe in a lot of detail how that works, but here's an illustration of kind of on a toy problem what it looks like. So I mentioned how for each one of these linkage maps we generate this list of inequalities, and the inequalities are capturing the relative order of the markers in each one of the maps. So here I took just a dummy toy problem. This is a set of 7 markers. I just put them positions 0 through 6 on the whole numbers. And then I took a set of markers and I just flipped them to represent 3 other populations which have a very similar linkage map, but there's a small error in the order. So in population 2, I flipped the order of B and C, F and G, so on. Okay, so if you look at the inequalities, you'll see that, like in population 1, for example, it says that the position of marker C has to be greater than or equal to the position of marker B, but it's the exact opposite in population 2. It says that B has to be greater than or equal to C. Well, the only solution to that is that B and C are both identically equal to 0. If B has to be greater than or equal to C and the converse, then basically you're trapped into this only solution being that all the markers collapse down to 1 point. Okay, you don't get a consensus map out of it. It's not a useful solution. So what we have to do is we have to find a way to identify which inequalities are in conflict and remove them so that we can get a non-trivial consensus map out of this. And it turns out when you study what that looks like mathematically, it's been studied by computer scientists, and it goes by this name of the maximum feasible subsystem problem, which basically is, in layman's terms, saying you have a bunch of inequalities, you want to remove the fewest number of them before fewest number, remove them so that you can have these non-trivial maps coming out of it. And we know that that's computationally difficult if you're trying to find the exact solution, but you can get pretty good approximations with linear programming, as it turns out. So linear programming is used both in the error minimization and in this application of resolving conflicts. And just published last week the details of how this is done in bioinformatics, so you can check it out if you're interested. All right, here's what the results look like when you're running on this toy problem. LPmerge exactly identifies the exact 3 places where I just tweaked with the map. It said the output from the software says, oh, I'm going to remove the inequality that C is less than B from map 2. I'm going to remove the inequality that G has to be less than or equal to F from map 3, and likewise for map 4. So all the places where I just kind of tweaked it, it is able to use the consensus information to identify which inequality has to be dropped. And the output from that is actually you recover the exact correct toy map in this situation. So that was a nice demonstration of it on a toy problem. How does it work in practice? Well, going back to Barley again, there was a new Barley chip that was developed in the last couple of years which had 9,000 markers instead of 3,000. So there was an old consensus map that had 10 populations. I talked about 4. There were then 6 more to make 10. And then they wanted to add 1 more population because they had genotyped it with the 9,000 array. And actually, we tried to use merge map on this problem, and it crashed for reasons I don't understand. But the LPmerge software worked quite well. It was able to identify 12 conflicts in order and resolve them. And the consensus map has 5,600 SNPs. And this has now been used by several groups to do some GWAS in a USDA Barley core collection. This is really work that was done by my colleagues, Maria Munoz-Amitron and Alfonso Cuesta Marcos. Just to show you, okay, typical GWAS results, they're able to use the consensus map to study a number of morphological traits in Barley. So just to conclude, this is a new software. It's available. It's based on linear programming to both minimize the error between the linkage map and the consensus maps and to try and resolve these order conflicts. And it's not just Barley, but people are using it for pine, wheat, cassava, and having good success. So if this is an application you're interested in, you can find out more about it at my website. It's also on CRAN. So I encourage you to check it out and let me know how it works for you. Thank you.
Thank you, Jeffrey. That was a very clear and nice presentation on a difficult subject for some of us, but I know I've used MergeMap myself and had it work fine, but I think you have something that's quite a bit better, so congratulations on that. I think we have time for maybe 1 or 2 questions before coffee. Any questions?
Oh, hi, it's a nice presentation. Just I want to have clarification that join map also allows such modifications, and then you can work on based on recombination, whether it's like markers are distorted, your loci. If you're confident on your framework map, you can choose one as a framework map. So something I want to hear that do we have such comparative options here as well?
I heard you were describing a bit about join map. What was the question? You wanted to know whether you could do what? With
Yes. So what were options available with join map? Whether we have such options in LPmerge map also?
Well, join map is certainly a more professional software package than what this is going to offer you. I think the experience that a number of people have had with join map is that when you have a lot of populations, the running time can be quite large. Maybe you've had a different experience, but this is really designed once you've created linkage maps for each population in join map, for example. This won't generate genetic maps within a population. It just merges them, and it has some diagnostic features, but certainly nothing like what the Windows interface that join map would have for you.
Great, thanks. Andres?
First, do you select a seed map for consensus building? And the other one, do you think that this error detection can be applied also to biological inconsistency? As I said in my presentation, we're facing this phenomenal structural diversity in wheat mapping and also any polyploid maps. And do you think that you can use the same approach in sort of getting good consensus in spite of this structural diversity?
I do think the first question, is there a seed map used? No, that's not intrinsic to the way this works. And I think the ability to identify, the output really is, it doesn't identify spurious markers, it identifies marker relations, like orders that aren't consistent with others. And if those are of a biological nature, then perhaps it will provide insight as to where there are underlying biological differences as opposed to just numerical artifacts. I'd be interested to hear how it works if you would try that. Yeah, the output, it lists for each map which of the inequalities it finds that it's trying to remove to create the consensus.
Maybe we have one more burning question. Yes, back there.
Hi. I just wanted to say that it's pretty important to take out, when you've got loads and loads of markers on your map, it's an important thing to take out the bad ones before you merge and make a consensus map. So an alternative program I would just like to point out to join map is thread mapper. I mean, people might be familiar with it, and it's a good one to use. Get rid of the junk markers, then merge using your program or another one.
Yeah, it's a great point. The quality of your consensus map is only going to be as good as the quality of the linkage maps that you give to it. So it's important to do that type of quality control.
Great. Well, thank you again, Jeffrey. That was very, very nice presentation.